Now guys, it's part 2 of chapter number 3, open state versus closed state of a gated channel. Note that the channel conducts current in all or none fashion. What it means? It means that at a certain voltage the channel will remain open and will conduct the current, and at another voltage it will remain closed and it will not conduct the current. So desirably, if we change the voltage to a certain point that the channel remain closed, then it will conduct the current continuously until the voltage is changed. And if we bring the voltage to a certain point that the channel will remain closed, it will remain closed until the voltage is again changed. So the channel will remain closed or it will remain open. It will conduct the current or it will not conduct the current. Page clamp method for recording ion current flow through the single channel. What is the page clamp method? Page clamp method, first of all, we will abut it the micropipette against the cell membrane. We will abut it this micropipette against the cell membrane. To cut a piece of this cell membrane is a page, and then we will seal this micropipette with this page on this side, and then we will change the ionic distribution and composition inside the channel, and we can also change the ionic distribution and composition outside in this water bucket, and we can and through these changes we can study the conduction of current through a particular channel. And through this page clamp method, even we can study the conduction of current through a single channel by changing the voltage and ionic distribution outside the micropipette and inside the micropipette. So this is called the page clamp method. Now, the facilitated diffusion requires membrane carrier protein. We have diffusion, for example, simple diffusion and facilitated diffusion. Simple diffusion, as we have discussed in the start of this chapter, that it may occur through the lipid bilayer or it may occur through the protein pores or channels. Facilitated diffusion occur with the help of carrier protein. For example, if this is the cell membrane, this is the carrier protein, this is the receptor. The substance will attach to the receptor, and this substance will change its conformation and will take up the substance into the cytoplasm. This is the facilitated diffusion. They have discussed another point in this diagram. For example, they have discussed that with the increasing concentration, the simple diffusion increases without any interruption and is directly proportional to the rate of the simple diffusion. The concentration is directly proportional to the rate. But in case of the facilitated diffusion increases with the increasing in concentration, but it reaches to a certain point where it remains constant. Why the facilitated diffusion comes to a constant value? Why? This we will explain this. For example, if this is the maximum value, 100 is the maximum value for the carrier protein to change its conformation 3 times per minute. If we change the molecules outside the cell membrane to its maximum value, which is 100, for example, then at this maximum value, the carrier protein will change its conformation 3 times per minute. But if we exceed this value to 200, for example, if we increase the molecules to 200 molecules, then can it will be able to change the conformation? No, because it's the maximum capability of the carrier protein to change 3 times per minute, to change its conformation for these molecules 3 times per minute, and this maximum capability of changing occurs at 100. So if we increase the molecules to 200, it will not affect the capability of this carrier protein to change. So that's why the maximum point occurs for the facilitated diffusion. This is the reason. Now, facilitated diffusion is done. Now we will come to the factors. Factors that affect the net rate of diffusion. Factors that affect the net rate of diffusion. Net diffusion is directly proportional to the concentration difference across a membrane. For example, in this diagram, this is the cell membrane, this is the pores, This is the inside and outside. These are the molecules which we have discussed the mechanism of the diffusion, that how the diffusion occur. Now we will apply that mechanism in this diagram. The molecules and the gases and the liquids are in constant motion, so they are moving constantly and striking each other in the cell membrane. So on the side where the molecule is more, the striking will be more and they will apply greater pressure. On the inside, the molecules are also striking each other and they also produce spores on the inside and applying pressure. But the striking pores and the striking pressure of these molecules is less as compared to this. So more molecule will diffuse to the inside and less molecule will diffuse from the inside to the outside. So we will take, we can say that the diffusion is directly proportional to the concentration difference between the two sides. What it means? It means that we will minus the concentration which is inside from the outside. So we can write this diffusion is directly proportional to the concentration difference. Concentration difference means the concentration which is outside, concentration outside minus concentration inside. So that this is that net diffusion rate is directly proportional to the concentration difference across a membrane. Now diffusion also rely on the membrane electrical potential and diffusion of ions, the nascent potential. We will discuss this. For example, if the cell membrane is negatively charged on the outside and positively charged on the inside, and there are molecules which are negatively charged, so these negatively charged molecules will be attracted by the positively charged molecules which are on the inside of the membrane. So they will be attracted to the inside, and through these electrical attraction, the concentration of the on the inside of the cell membrane will reaches to a point that these concentration will produce its pressure, and this pressure will equalize the attractive force and will cancel each other. For example, if the concentration is attracting to the inside and the inside concentration is increasing until it reaches to a point where the concentration will stop further diffusion of ions. Why? Because the attractive force of the cell membrane will be neutralized by the pressure which is produced by this high concentration. So this is the membrane electrical potential and diffusion of ions. How the membrane electrical potential affect the diffusion. Now, effect of a pressure difference across the membrane. What is the effect of pressure? For example, this is a piston. If we apply a little bit of pressure, then there are the same molecules on same molecules on inside and outside of the piston. But if we apply the pressure, then the pressure will increase on this side and they will strike the membrane of this piston with high velocity and will diffuse to the outside of the piston. So in this way the pressure also affect the diffusion. Now the diffusion is affected by the concentration. When the concentration, the diffusion can also be affected by the electrical charge, and the diffusion can also be affected by the pressure. So these three main things can affect the diffusion. Now osmosis across selectively permeable membrane, net diffusion of water. For example, we have noticed that in each second there is the exchange of water through the red blood cell is 100 times as compared to its volume. So when the water is entering, then immediately it is coming out of the cell. So the exchange of this water, how much of the water is entering and how much is coming outside, this is equal. When 1 liter when 1 microliter is entering to the red blood cell, then 1 microliter is coming out. So the net rate of diffusion is 0. But if this net rate of diffusion is disturbed, if the net rate of diffusion, for example, if the diffusion of water is increased to 2 microliter and the outcoming water is 1 microliter, so when this net rate of diffusion is disturbed, then this net rate of diffusion through a selectively permeable membrane is called osmosis of water. If the water is entering and outing, the incoming of water and outcoming of water is equalized, then the net rate of diffusion is zero and it cannot be called as osmosis. If the net rate of diffusion is disturbed, if more water is coming or more water is coming in or more water is coming outside of the red blood cell, then it can be called as the osmosis of the cell. In simple word, the net rate of diffusion, the diffusion across a selectively permeable membrane is called osmosis of water. This process of net movement of water caused by the concentration difference of water is called osmosis. Now we will discuss the mechanism of osmosis. For example, if this is the sodium chloride solution which also contain water but in less quantity, and this is the pure water, so the motion of the molecules of water will be more on this side, and the motion of molecules of water in this side is less because the quantity of water is less in this side. So the striking force of these molecules produce a pressure which diffuse the water from this side to this side. Why? Because the water pressure on this side is less as compared to this side. So the water will diffuse from low pressure to from high pressure to a low pressure. So this is the mechanism of osmosis. Now the osmotic pressure. So the amount of pressure required to stop osmosis is called the osmotic pressure of the sodium chloride solution. For example, in this figure, they have explained when we put pure water on this side, and in this side contain sodium chloride solution, and these two are separated by a barrier. So the water molecules are more, so they will produce more pressure on this side, and on this side the pressure will be less for water. So the water will start diffusing to this side and will increase the level of this side until it will reaches to the point until it is prevented by the atmospheric pressure for to rise further. So this pressure which has stopped this osmosis, this is called osmotic pressure. For example, the water is continuously moving to this low concentration, to this low pressure area, and this low pressure area is continuously rising until the atmospheric pressure is so enough that it can stop this osmosis. Then this pressure is called osmotic pressure. So this was the osmotic pressure. Now, importance of number of osmotic particles in determining osmotic pressure. The osmotic pressure is determined by the number of molecules, not by the size of molecules. Why? Because the smaller the molecule, the high will be the velocity, and vice versa. If the molecule is large, then its velocity will be low. So that's why the kinetic energy is more in smaller molecules and is less in large molecules, and they produce the same the same pressure inside. So that's why the number of particles is important for producing the osmotic pressure, and the size of the molecule does not affect the osmosis. Osmolality. What is osmolality? Now I'll show you what is osmolality. The number of particles per kilogram of water is called osmolality. Now, for example, if we take some examples for your understanding, 1 mole of glucose is equal to 180 gram of glucose, and this glucose cannot disperse into separate particles in a solution. So the osmolality of that solution which contain 1 mole of glucose will be 1. But if we take 1 mole of sodium chloride, then and if we take this in a solution, the sodium chloride will dissolve into 2, sodium plus chloride. So the osmolality of that solution will be equal to 2. Osmolality will be 2. So it doesn't depend on the size of molecule, but it depends on the osmolality depends on the number of particles in a solution. This is 1 osmol. Suppose 1 osmol. The osmolality of this glucose solution will be 1 osmol, and the osmolality of this sodium chloride solution will be 2 osmol. So 1 osmol in our body is equal to how many? This is equal to 19,300 millimeter of mercury pressure. Then 1 milliosmol will be equal to 1 milliosmol will be equal to millimeter of mercury. 1 osmol will and 1 osmol is equal to how many pressure? It is equal to 19,300 millimeter of mercury, and 1 milliosmol will be equal to 19.3 millimeter of mercury. But our body, the osmolality of the extracellular fluid of the human body is about 300 milliosmol. 300 milliosmol. So the pressure in our body will be equal to 750 millimeter of mercury. Our average pressure in the human body is equal to 550 millimeter of mercury. So with the help of this osmolality, we can calculate the extracellular fluid pressure. Now what is osmolality? There is a little bit difference between osmolality and osmolarity. The number of particles per kilogram of water is called osmolality, and the number of particles per liter, this is called osmolarity. The number of particles per liter called osmolarity. The number of particles per liter is called osmolarity. And now in the daily using of the osmolarity and osmolarity, the osmolarity is more usable. So this was the difference between the osmolarity and osmolality, and the osmolarity and the osmotic pressure depends on the separation of the molecule and on the numbers of molecule, not on the size of the molecule. For example, glucose and sodium. This is 1 osmol, this is the 2 osmolar solution, this is 1 osmolal, and this is 2 osmolal solution. 1 osmol is equal to 19,300 millimeter of mercury, 1 milliosmol is equal to 19.3. Our body fluid has osmolality of 300, so we have 750 millimeter of mercury pressure. So this was all about the osmolarity and osmolarity. This is the 500. This one more. You should add one more zero. This is 5,500 millimeter of mercury is the average pressure of our extracellular fluid. Now the second part of this chapter is finished. Please guys support my channel, like, subscribe and comment. Thank you.